Hello everyone, my name is Siti Hajar binti Abu Samah and I will present my final year project which is effective microorganism marble for water quality treatment in river. For introduction, rivers have many importance for human beings such as agriculture, sanitation, generate electricity, food source and transportation. The river water quality degrading along the time due to urbanization and industrialization cause limitation for the river function. Awareness for the clean river is increasing due to clean water shortage, which is related with sixth goal of Sustainable Development Goals by UN. And scientists, engineers, and researchers have been tried to find solution either chemical and biological treatment. So, what is effective microorganism? It is made of yeast, lactic acid bacteria, and phototropic or photosynthetic bacteria to act as bioremediation to treat the river water. EM founded by Professor Teruo Higa in 1994 for agricultural purposes in the beginning, and effective microorganism improve microbial population by fermenting the organic matter in the soil. In water treatment, the EM breaks down all the organic matter and sludge and other beneficial bacteria feed on the decomposed sludge. And EM1 is the base product while EM activated solution is the mixture of the molasses with EM1. However, it does not last long than the base product. Problem statements state that the river quality have been degraded and it may achieve to the worrying level where the river resources becomes more polluted until it is not safely consumed to the human and animal. For the reduction of water pollution level, various chemical and biological treatments are available, and the emergence of an amazing technology of a multicultural of anaerobic and aerobic beneficial microorganisms is presently gaining popularity due to its environmentally friendly nature. However, the microbiome is not enough to treat the whole size of the river thoroughly since river can be flowy. So, we introduce EM marble. So, what is EM marble? So, EM marble is consists of clay soil, bokashi component which is conversion of food waste into soil amendment by compost fermentation, and EM with molasses for activating the EM. So, why we are using marble? This is because the marble gives weight to the EM to reach the lower level of the river. Hence, the effective microorganism could decompose the sludge at lower level and clean the river thoroughly. So, for the research objective, we are going to find out the effectiveness of EM marble using different materials and also to characterize EM marble and compare from the previous study. For the literature review, we have different types of application which have been studied from previous research. EM have not only treating the river but also to treat heavy metal in textile industry, domestic sewage and grey water too. Also, EM have been experimented with different kind of EM marble material. This shows that EM is a versatile bioremediation method which could help us to fix the environment to be better. For the methodology, this is our flowchart for the method, and we will start by finding the preparation of EM marble from the previous study, and also we find the parameter measurement that involved to measure the water quality. Next, we find the characteristic of EM marble to achieve the objective 2, and then we are using critical review to make a result and discussion for this research. And also lastly, we are using water quality index to measure the water quality. So for the first methodology, we are preparing the EM marble. So for the first material, we are using 14 kg of soil clay, 700 g of EM bokashi, molasses, which is called as EMAS, which is effective microorganism activated solution, and also additional other ways such as rice bran, fruit branch, and more. So for the procedure, we will mix all the clay soil and bokashi component in the deep tray Pour the activated EM until the mixture can be rolled. Roll the mixture until it becomes a size of tennis ball and put it in container that have newspaper at the bottom. Next, the container need to be kept out of direct sunlight or placed with strong wind to prevent the marble dry too fast. As the marble are covered with the white fungus on the surface, the marble are ready to be used. As for the parameter involved, we have total solid suspended, biochemical oxygen demand, ammoniacal nitrogen, pH, chemical oxygen demand, and dissolved oxygen. To achieve objective 2, we have characteristic of marble. So, different from the common marble, from the procedure of preparing EM marble, it will become white mold due to fermentation process occurring in the marble. The marbles should be solid after fermenting and drying and not fall apart so easily. Do not let the marble become too hard as it takes a while to soak up water and sink to where they are most effective. According to EMRO Japan, the white mold helps for the beneficial microorganism to grow and then to help the release of the infection. The next methodology is the critical review. Most of the articles and journals show that in situ experiment and laboratory analysis are commonly used in this research. In situ experiment involve at the experimental site, which is to collect sample from various points. The sampling station is made with 6 different places, which is determined upstream, middle, and downstream. Laboratory analysis is needed to measure each parameter involved in determining the river quality. The measurement unit is in milligram per liter. Each parameter result will be compared with water quality index to determine the river classes. So this is water quality index, which is sourced from Department of Environmental, DOE. So we can see here we have different classes that shows every parameter, and then different class that differentiate the type of river quality. So for the first result and discussions, we are comparing Ahmad et al. 2018 and Wahida et al. 2016 because they have same method and location to be used in carrying the experiment. Both are also running laboratory analysis to run the test and in situ Sungai Sebulung area to collect the sample. For this journal, the WQI of the river was 56.2 and classified as class 3. And the low WQI index can be attributed to high AN and slightly low concentration of DO. During low flow of river water, DO level did not increase since some of the weirs are higher than the water level. This caused the water cannot flow continuously. So from the graph, it shows the average of concentration for WQI parameters against sampling stations at Sungai Sebulung. From the study of Wahida et al. 2016, each graph are the result from this journal and each parameter from different sampling station, which include BOD, COD, AN, TSS, and DO. From overall result, the data to water quality index, the overall class of this river also improved from class 5 to class 4. Only several parameters that EM did not manage to reduce, which are dissolved oxygen and total suspended solid. So, for the critical review of previous study, first we are looking for dissolved oxygen. So here, we see that the optimum DO reached for the first research is 3.59 and the second research is 6.30. From this both study, DO expected to be less than 10 mg per liter because of the EM1 solution effect in the marble. However, the DO shows increment from the sample that shows before using EM. Hence, according to the study, rice water causes the DO increase in smaller rate. For the pH, we are comparing 3 journals, which shows the optimum pH within the range. For the good quality river, it is in the range of 5 to 7 approximately. The increment of BOD, COD, and TSS could affect the pH. A rise in pH is a proof of the bacteria using up carbon dioxide during photosynthesis. Next is the total solid suspended. So we have two journals here that we compared by using different marble material, and shows that the first journal used 19.89 milligram per liter, and also the second journal have 100 milligram per liter. So expected optimum TSS for both study is 25 to 50 milligram per liter. So here, the second journal exceed the level of TSS. Hence, according to Hika et al., EM were used to reduce content of wastewater by 94%. This shows that TSS removal efficiency increases by using more EM marble quantity, as the absorption of TSS will be better. Finally is the ammoniacal nitrogen, which compared by using two references. However, Wahida and Azman exceed the range of optimum AN, because in this study, AN expectedly to be in range of 0.3 to 0.9 milligram per liter. Can be affected by the usage of excess chemical fertilizers nearby the river of Sungai Sebulung, Johor. So, in conclusion, based on the previous study, there was significant different or decreasing parameter of pH, temperature, TSS, and AN. The O value increment also indicate the treatment is considered as successful. BOD and COD can be factors that influence the river quality, which is needed to be studied in future. And for other recommendation, future experimental about EM marble should be done in laboratory for accurate analysis. That's all from me. Thank you.